Hello guys, my name is Isaac, simple and real. Thank you for tuning into my channel. Kindly, if you have not yet subscribed to my channel, please subscribe, like and share. This is a channel which talk about issues concerning averting HIV and sexually transmitted infection stigma in our society. And we usually see that being positive about HIV is not necessarily being HIV positive. Thank you. Today, I want to talk about the life cycle of HIV virus. And what's the rationale of doing this? Basically, this is to help any person who is listening to me to be able to understand why or the rationale or the importance of taking the medication appropriately and its impact into the immunity of human, or for us to understand the role of virus in suppressing our immunity when someone is HIV positive. Let's learn. Basically, I will use a very simple procedure in describing this. A HIV virus life cycle entails 6 phases. Phase 1 involves the reception of the HIV virus into the CD4 T lymphocyte cell, which is part of human body immune cells. Now, let me try to explain so that you may be able to understand this process. A HIV cell contains projections, or which we call glycoproteins. It also contains an envelope, and it is also an RNA virus. We have 2 types of viruses. We have DNA viruses and we have RNA viruses. Now, HIV is an RNA virus. On the other side of human body, we have various immune cells. One of the immune cells we have are called CD4 T lymphocyte cells. Now, why do we talk about CD4 T lymphocyte cells when we talk about HIV virus? This is because human CD4 T lymphocyte cells have got receptors and parareceptors which accommodates the HIV virus glycoprotein to form an invagination. Are we together up to this point? Thank you. Now, when the HIV virus invaginates itself into the CD4 T lymphocyte receptors and parareceptors, we move to the second phase. Second phase entails reverse transcription. Now, after the invagination, the virus releases an enzyme called reverse transcriptase enzyme. The role of this enzyme is to enhance the conversion of the RNA strand inside the HIV virus into a DNA strand. Usually, RNA is a single strand and DNA is a double strand. Now, when HIV virus RNA is now converted to a DNA strand in presence of reverse transcriptase enzyme. From there, we move to the third phase. Now, the third phase entails the integration of now this DNA virus strand into all this. We have 2 types of now the DNA strands. Remember, a human cell nucleus has got a DNA strand, and now this virus has got a DNA strand. For them to be integrated together, we need an enzyme. And this enzyme is produced by the virus, and we call it enzyme integrase, which integrates virus DNA into human DNA. Now, when this virus DNA is integrated into human DNA strands, we call this DNA a provirus DNA. Now, a provirus DNA cannot be detected as foreign by the human immune system. So this provirus DNA is now protected from attacks by the human immune system. From this point, we move to the fourth phase. Now, the fourth phase of the life cycle, we call it the transcription phase. Now, what happens in the transcription phase? Usually, the host cell, which is now the CD4 T lymphocyte cell, releases an enzyme which we call it a polymerase enzyme, or we call it RNA polymerase enzyme. Now, what's the role of RNA polymerase enzyme? Now, the role of this RNA polymerase enzyme is to break down the provirus DNA into a very long chain of messenger RNA proteins. Are we together up to this point? Thank you. Now, from this point of transcription, we go to the next phase, which we call the assembly phase. During the assembly phase, the virus releases another enzyme, which we call protease enzyme. Now, the protease enzyme helps in the breakdown of the long chain of messenger RNA proteins into short chains messenger RNA proteins. Also, during this phase, the RNA strand is combined with now these short chains of messenger RNA proteins to form a virus particle. Are we together up to this point? Thank you. Now, we have now formed a virus particle from an RNA strand and short chains of messenger RNA particles or proteins. Now, when we form the virus particle, remember this virus particle is still inside CD4 T lymphocyte cell. Now, we move to the next phase. Now, this phase is the sixth and the last phase, which we call budding phase. Now, the virus particle starts forming buds. Now, these buds that have started developing, they destabilize this host cell. Now, when the host cell dies due to the destabilization, when this virus particle starts forming buds or die a natural death of a cell, the virus particle obtains the membrane or the cell membrane of this CD4 T lymphocyte cell to form its envelope. Now, we had the viral proteins, and now the viral proteins, after this cell has died, they form the projections on the envelope, which we call them the viral glycoprotein. And in this process, a new viral cell has been formed. Now, this cell can now attack another CD4 T lymphocyte cell to form another host, and the process continues. Now, something I had not said, this virus particle is usually not one of them inside a host cell. They are several, and in this process we can be able to release even a big number of viral cells which have been formed into the system or into the blood system for them to attack other T lymphocyte cells and attach to them, and the cycle continues. So in the process of one T lymphocyte dead, we raise several numbers of viral cells. Are we together up to this point? And this is the reason why, as you continue not to take your antiretroviral drugs, the viruses continue attacking more T lymphocyte CD4 cells, and your CD4 go down because CD4 cells are dying while the viruses are multiplying. And if you do not take good care of your CD4 T lymphocyte cell, they may end up going so low to an extent that you might not even be able to fight even small infections that are rather or your body could rather clear them without your intervention with drugs. And these are the people, this is why we call HIV virus an immunosuppressive disease. Are we together up to this point? Thank you. So guys, this is the main role why you should be able to take your ARVs to prevent the replication of the virus inside your body, so that you can be able to save the remaining CD4 cell as your body continues to form more or to generate more CD4 T lymphocyte cells to fight or to protect your body, or so that you can be able to maintain the integrity of your immune system. Are we together up to this point? Thank you guys. Thank you even for watching up to this point. Kindly, if you have not yet subscribed, please subscribe and also share so that other people can learn from this. Thank you.